Thanks very much for coming. My name's Tony David. I'm the Vice Dean for Psychiatry here at the IOPPN. It's a great pleasure to welcome our speaker today, Dr. Anissa Abi-Dargham. She's the 16th Paul Janssen lecturer. Let me just tell you a little bit about her. She is Professor of Psychiatry and Radiology, a combination we don't have here at the Columbia University Medical Center, and she's Director of Translational Research at the New York State Psychiatric Institute. So Anissa did her medical qualification at the St. Joseph's University, which has a branch in Beirut, Lebanon, and then did her internships in the US, did her residency training in psychiatry in Tennessee. Must have been a bit of a culture shock. Then fellowships at Yale University, and then I think after that has been at Columbia and has been pursuing an incredibly productive research career ever since. So Anissa is also the president-elect for the American College of Neuropsychopharmacology. She's deputy editor of biological psychiatry and associate editor of neuropsychopharmacology. Is on important boards for the National Institute of Mental Health and NARSAD and many other honors. She is a pioneer in schizophrenia research, and as you'll hear, has really firmed down and proved the importance of dopamine in schizophrenia. Interestingly, of our many publications, her most cited paper is a study about amphetamine-induced dopamine release. That's nearly 1,000 citations. And that ties in very well with the Paul Janssen lecture, because, of course, Paul Janssen was a pharmacologist, and it's said that he developed haloperidol, one of the most important early antipsychotic medications, when working in Belgium with and coming across professional cyclists who were going into the Tour de France and were using and abusing amphetamine and becoming at times psychotic. And so he developed specifically haloperidol as some antagonist to the effects of amphetamine. I don't know whether it was really understood that dopamine was the key ingredient there, but clearly that's been a landmark in the field. And then taking that further and proving the importance of dopamine has really been down to Anissa's work. When I've done introductions for this lecture before, I've found it quite useful and amusing to go through speakers' publications, and then you usually find a few sort of comment pieces and opinion pieces where they're slightly off guard and they make statements that I can then feed back. Not so. Anissa's too focused, too professional. All the work is extremely high quality science. But there is one paper that stands out, and it's the title that stands out, and it's called Dopamine as the Wind of the Psychotic Fire. And this has been picked up by Oliver House in his publications and Satish Kapoor. And it is something very poetic, something out of the Rubaiyat of Omar Khayyam, perhaps. And there's no explanation where that comes from, but I suppose it does suggest an underlying interest in metaphor and poetry as well as science. So I hope we're going to hear a little bit about that today in the lecture. I should say also that this being the 16th Paul Janssen lecture, I think it's the first time that we've had a woman scientist, which is a great shame on us, but hopefully it's going to be the first of very many. So Anissa, welcome, and we look forward to your lecture.
Thank you very much for the nice introduction and also for the very special invitation. It's a true honor to be here. First, to be at the Institute of Psychiatry, where so many researchers are doing work that I find extremely admirable. and very close to my interests, and also to be a Paul Janssen lecturer, especially that Paul Janssen's line of work in the discovery of Haldol is one of the pillar elements of the story that I've been interested in during my academic career. And you will see some of the work that is really kind of evolved from discoveries like this. It's also very special to be in London, such an interesting city, and I've had a small taste of it, definitely enough to have to want to come back. But thank you so much for this honor. And I'm really also glad to be the first, the inaugural female scientist, Paul Janssen lecturer, and I definitely hope you'll have many more female scientists speaking here. So what I'm going to do today is summarize to you with a little bit of a kind of not into the detail, but more the big picture of what we have found in studying using PET imaging to study dopamine function in schizophrenia. First, my disclosures. And then I'm going to start with a little bit of a historical perspective. You can hear me okay? Okay. So the historical perspective on dopamine and schizophrenia. So it all started with the discovery of the antipsychotic properties of chlorpromazine by Delay and Deniker in 1952. And this was very much closely followed by the discovery of haloperidol in the lab of Paul Janssen in 1958. Then the finding of Carlson that dopamine turnover is increased by antipsychotic was kind of one clue incriminating dopamine transmission in psychosis. And the first formulation of an excess of dopamine in schizophrenia was really put forth by Ventralstriatum in 1966. This was followed by the discovery of dopamine-sensitive adenylyl cyclase in the lab of Paul Greengard, and then by the discovery of that antipsychotics have binding sites in the brain by Snyder and Seaman in 1976. And then the cloning of the D1 and D2 receptors. And later, more into the 80s, the discovery that dopamine agonists do the opposite of these D2 antagonist drugs in the sense that they increase psychosis while D2 antagonists block psychosis. So this evolved pretty much into the first formulation of a dopamine excess, targeting mostly what was talked about in those days was the mesolimbic system was overactive, producing psychosis of schizophrenia. So this was followed by a reformulation in the late 80s, early 90s by Danny Weinberger and Ken Davis, proposing there is actually a dual imbalance, that is not just excess in the striatum or subcortically of dopamine, but there is also a deficit in cortical dopamine because they had to somehow work into this theory the fact that dopamine mediates prefrontal cognition and that prefrontal cognition is deficient in schizophrenia. So one explanation would be that in the cortex is actually a deficit in dopamine. There was even a concept of causality between these two where Danny Weinberger proposed that cortical deficit may induce subcortical excess of dopamine by way of which dopamine modulates the circuitry that regulates subcortical dopamine. And so what I'm going to propose today, or at least just suggest, is that we may be at a point where enough work has been done now from our lab and other labs like Oliver House's lab that put us at a point of a possible reformulation. So this would be the third formulation, because we have evidence supporting the dual phenotype, although it needs confirmation. We also have put on the map a structure that wasn't incriminated earlier on, which is the associative striatum, as opposed to the ventral striatum or limbic striatum. And we have extended the concept of deficit to regions in the brain that were never suspected to be deficient in dopamine. Basically structures that people didn't think about as being affected by hypodopaminergia, as in the cortex. So I'm going to show you the data supporting these claims I'm making, or these questions that I'm raising. Okay, so first let me just kind of, for people in the audience who don't necessarily think about dopamine constantly, just take you a little bit, like some of us are obsessed with dopamine. So the dopaminergic pathways have their cell bodies in the midbrain region of the brain, and they are organized in layers. So you have the ventral tegmental area cells, and then you have cells within the substantia nigra, compacta, and reticulata. And the compacta has a dorsal tier and a more ventral tier. And this topography is relevant as to where are the dopamine cells projecting to which parts of the brain, so that the VTA and the dorsal part of the nigra compacta project to the ventral striatum, that's the mesolimbic pathway, and to the cortex, that's the mesocortical pathway, while the other cells that are in the deeper layers of the compacta and the reticulata project respectively to the associative striatum and to the sensory motor striatum. This is borrowed from Suzanne Haber, and the color is coded so that red indicates regions that are involved in processing emotional information. Green and yellow is cognitive, associative type of information, and blue is sensory motor information. So these dopamine cells have different properties, where dopamine cells that are in the VTA exhibit what is we call burst firing. So they fire in very rapid segments where you have few spikes very shortly within close to each other. And that is mediated by stimulation of the dopaminergic cell via the NMDA receptor at the level of the midbrain. That just kind of excites them so that they burst fire. And this is mostly a property of VTA neurons, while the dopamine neurons of the substantia nigra don't show as much of this burst firing. They have more of a tonic firing that is driven by the tonic GABAergic input onto them, so keeping them basically under tonic inhibition, so they're allowed only very slow firing. And it's also regulated by potassium channel SK3 and calcium channels, as well as the D2 receptors. So these channels basically work to just decrease the excitability very quickly to control and keep those dopamine cells from staying excited. And this is illustrated well in this very nice review that I suggest for people interested in this topic to read, where Jock and Roper puts all the data together and showing the phasic firing that you can see in mesocortical dopamine neurons and mesolimbic dopamine neurons, while you have the tonic firing and the other pathways, the mesostriatal and the lateral shell of the mesolimbic. So now you have to understand that phasic firing is very important to drive learning. This is basically what happens during cognitive tasks or in response to reward or things that kind of drive behavior. So that's an important property of the mesolimbic and mesocortical pathway. Another important kind of organizing piece of information about the role of dopamine in the brain is, so we've covered where are the dopaminergic nerve cells and how they project to those brain regions. But then the output from those brain regions that is kind of centralized in the striatum and going back through the basal ganglia loop back to the cortex is also very much under the influence of dopaminergic receptors. So you see here, and this is a very famous slide from Gerfen and Surmeyer, showing the glutamatergic pyramidal cells input onto the striatum. You also have input from the thalamus, of course. All these are glutamatergic. From the striatum, and you have like there in red and blue or the D1 and D2 receptors. So the output from the striatum is carried by the medium spiny neuron. And these are subdivided into two types, one that has D1 and one that has D2. And the D1 pathway is called also the direct pathway or the GO pathway, while the indirect pathway that has D2 receptors is the no-go pathway. And the reason they're called this way is because of their behavioral effects. So the D1 pathway is such that it facilitates the thalamus speaking back to the cortex. The way it works, you have this GABAergic projection onto the globus pallidum internal, the substantia nigra reticulata, which are the basal ganglia output nuclei. So you have an inhibition onto another inhibition at both levels, which basically results in facilitating the thalamus feeding back to the cortex. So that's kind of the loop that allows us to say, okay, go, approach, that's good, go ahead. While the indirect pathway does the opposite. It's the way the circuitry works is that it's the inhibitory pathway where it tells us to avoid, stop, do not do something. And it's the reason is that you have these kind of intermediate connections that makes it, so for example, you inhibit the inhibition onto the inhibition to the thalamus. So you basically inhibit the thalamus. And our behavior is organized according to a balance between this go and no-go pathway. And it's a complicated connectivity there that is very much under control of dopamine. Okay, so now with this introduction about dopamine, let me go into the imaging studies and what's been found from our work and also as a labs. So this is the synapse within the striatum, and I start with the striatum, then go to the cortex. Within the striatum, this is the presynaptic dopaminergic nerve terminal, so that dopamine pathway that goes up to the striatum. And we're looking here at the output of the striatum, which is the GABAergic medium spiny neuron that has either D2 or D1 receptors that we're talking about. So within the presynaptic machinery, you have all these different elements. So tyrosine transformed into dopa, transformed to dopamine, taken up by the vesicular monoamine transporter, packed into vesicles, can be from there released into the cytoplasm and into the synapse. The cytoplasm could be metabolized by the monoamine oxidase and the catecholomethyltransferase. In the synapse, it can be taken back up by the dopamine transporter, or it can interact with D2 receptors both presynaptically, and this is the autoreceptor, which senses the tone of dopamine and regulates the firing of this presynaptic dopaminergic nerve terminal to kind of keep homeostasis, or postsynaptically with the postsynaptic D2 receptor. And this has been offering multi-targets for imaging. And we've taken advantage of some of these. So obviously it's easy to design radio tracers that target the D2 receptors, and there are many of those. People also can look at the activity of the dopa decarboxylase, the amino acid dopa decarboxylase, which is what Oliver House has done very productively, and we'll see some of this work also here. And this is thought of as being an index of the activity of this enzyme, so the activity of the synthesis pathway and the capability for presynaptic storage. And then the other parameters that we've looked at is to image dopamine levels. Dopamine levels within the synapse at baseline by basically a subtraction paradigm where we remove dopamine, I'll show you, or the evoked release of dopamine through a stimulant challenge, which is basically how much is present in this presynaptic terminal that can be released after amphetamine. So these are the two paradigms we've used. We get a baseline binding potential, which is how much of this radio tracer, which is a radio tracer specific for D2 receptors, is binding in the striatum at baseline. Then we get a second measure, which is after dumping dopamine in the synapse via amphetamine or methylphenidate, which actually blocks the reuptake. The difference between these two tells us how much dopamine was stored that can be released. And the AMPT paradigm is kind of the mirror paradigm. We block dopamine synthesis for 48 hours by giving a blocker for tyrosine hydroxylase, so that it's long enough to allow all dopamine that's in the system to decay away and prevents new synthesis. So you actually have a brain that is dopamine free. So you get rid of all this dopamine, so now you can really see the true D2 receptors unmasked by dopamine. And by comparing these unmasked D2 to the baseline D2, where some of them were hidden by dopamine bound to them, then you get at how much dopamine was at baseline binding to D2 receptors, masking them, preventing the radio tracer from binding to them. So basically using all these, our labs and other people's labs have really converged in terms of what's going on with dopamine and the striatum, showing that the activity of this enzyme is increased, so suggesting there is increased synthesis of dopamine, and that's a lot of the work of Oliver House and Phil McGuire and other people. And that dopamine release as well as dopamine intrasynaptic levels are also increased. And the effect size is pretty much similar across the board, being close to 1. So it's a big dysregulation. This was summarized nicely in the recent meta-analysis by Oliver. So now we were interested to know, is it really the dopamine excess is in the mesolimbic pathway as proposed initially by the mesolimbic hypothesis of schizophrenia? So we created ways of subdividing the striatal substructures to isolate out the ventral from the associative in green, from the sensory motor in yellow, and this is by basically drawing regions of interest on these 3 different structures, anterior to the anterior commissure and posterior to the posterior commissure. So we can look at these separately. And we revisited some of the studies we had done that they had shown increased dopamine synaptic levels in the striatum, but now with the capability to isolate out these subregions. And what we found is, here you see in white controls, and in blue are the patients with schizophrenia. And you're looking at the removal of dopamine by the AMPT depletion paradigm. So the higher the numbers mean there was more dopamine at baseline. And you see that the biggest excess, the biggest dysregulation, was actually not in the ventral striatum, but in the precommissural caudate, part of the associative striatum. So that was the first initial, in a way, paradox. So this is not a mesolimbic excess in dopamine as much as it is an associative striatal or rostral caudate excess in dopamine. And that's almost more interesting and possibly makes more sense because the rostral caudate is the direct area of projection from the dorsolateral prefrontal cortex. It's also an area that gets a lot of inputs from limbic cortical regions. So it's kind of serving to integrate across 2 different functional domains. And it's kind of fertile grounds to think about aberrant salience and assigning meaning or significance to stimuli across different domains. But of course, this is speculative. But the interesting thing about this region also comes from the fact that we found that this is where the most exquisite relationship between dopamine release and psychosis happens. It's not as much in the ventral striatum as it is in the rostral caudate. This is the relationship at the level of the whole striatum between change in psychosis, increase in psychosis after the psychostimulant challenge, and how much dopamine is released by the psychostimulant challenge, right, using this amphetamine paradigm. So more dopamine release in the striatum, more psychosis. Revisiting the subdivision, this relationship was best in the rostral caudate and the associative striatum. So really the psychosis is kind of related to excess D2 stimulation in the structure. We also have described earlier that the more dopamine people have at baseline, the more likely they are to respond very drastically to D2 blockade, to antipsychotics, which makes sense. If they have psychosis because they have too much dopamine, then blocking D2 will help psychosis. And in terms of the ventral striatum, what we found is a different relationship altogether, which is the lowest they have, the lowest dopamine levels they have, the highest was the severity of their negative symptoms. So in the rostral caudate, too much dopamine psychosis, and in the ventral striatum, too little dopamine negative symptoms. While in the sensory motor striatum, we did not find a relationship to either negative symptoms or positive symptoms. Now, we may have possibly found a relationship to some other type of symptoms, such as motor symptoms, but typically we didn't look for that. But typically, patients that are in these imaging studies don't have too many motor symptoms. So now, another paradox that I want to bring up in summarizing this story is the following, which is that this study where we picked up the relationship between stimulation in the rostral caudate and psychosis was actually a study looking at dual diagnosis patients who have at the same time schizophrenia and addiction. Because they have addiction, they had lower presynaptic dopamine release. They had blunted dopamine, so their system is kind of affected by the addiction. And you know, a hallmark of addiction is low dopamine release in the striatum. That's what's been found across the board in alcoholism, cocaine, heroin. This is the work of Nora Volko, Diana Martinez, and some of our own work also on alcoholism. So if you have patients with schizophrenia that also have comorbid alcoholism, now their dopamine release looks like the alcoholics. It's down. It's blunted. But despite this blunting, this is the relationship that really, really puzzled us, which is their D2 receptors are super sensitive to any amount of small increase in dopamine that is released by our psychostimulant challenge so that it's psychotogenic. So that suggests that there is two things going on with the D2 system in schizophrenia and the rostral caudate, which is there's excess presynaptic release, but there's also a supersensitivity of D2 to stimulation that is contributing to the psychosis phenotype. So to summarize, looking at the striatum within like this whole circuitry, you have here the dopamine cells that project to different parts of the circuitry, amygdala, hippocampus, the cortex, the thalamus. The rostral caudate stimulation, excess stimulation of D2 receptors there by either excess dopamine or supersensitivity of D2 receptors is contributing to the psychosis phenotype, while what we have found, but needs further replication, is that low dopamine in the ventral striatum contributes to the negative symptoms. Now this is the landmark work of Oliver showing that in the high-risk group, they also have excess F-dopa, it's an index of dopamine synthesis, in the precommissural caudate and the rostral caudate. And this is the very early manifestation of the dopamine dysregulation, and it's predictive of conversion. And from there, it extends to other regions. So this, in a way, is a replication across different methods and different cohorts of the special role that the AST, the associative striatum, is playing in the genesis of psychosis. Now, he observed it in the high-risk phase. We don't know how much earlier it's present. And the earlier it's present, the more damaging probably it is to the circuitry, and the more negative are going to be the effects of that. Unfortunately, we can't study children to look at, people that are at risk even earlier to see what's going on there. But now we can kind of fill in the map, about the developmental trajectory. In terms of schizophrenia, you have the premorbid phase, the prodromal phase, when symptoms start to manifest, but they're not fully fledged to meet criteria. Then you have the first episode and deteriorations, and then the residual phase. And basically now we can clearly say, thanks to this work, that dopamine dysregulation starts in the prodromal phase and then gets worse at the acute onset. And then you have these kind of increases that we have demonstrated happen with acute exacerbations, more so that in between, when people are kind of in their quiescent interval of the disease, and probably less dysregulation during the chronic residual phase. So this kind of sums up dopamine in the striatum, brings up some of the important issues, role of the AST, the supersensitivity of D2. But now what about the cortex? Now remember, this is the reformulation suggesting that the cortex is maybe hypodopaminergic because the brain imaging studies show that it doesn't activate the way it should during working memory tasks, and also knowing how important dopamine is for D1 and D2 receptors in the prefrontal cortex to produce effective working memory performance. So we moved on to the cortex. We also had the kind of postmortem data from David Lewis's lab showing that in schizophrenia there is a decrease in tyrosine hydroxylase immunolabeling, suggesting either less innervation or possibly just a kind of malfunctioning of tyrosine hydroxylase itself. And since it is the rate-limiting step in dopamine synthesis, it would imply that there's less dopamine. So now, looking at dopamine per se was a very difficult thing to do, and it's been a challenge. So we started out by looking at D1 receptors. Now, D1 receptors were easy to measure with using just D1 radio tracers. Unfortunately, the data with the D1 imaging has been a little bit non-conclusive or conclusive in certain ways, but not in others. So using NNC112 radio tracer, which is a D1 antagonist that we also discovered had some affinity in vivo to the 2A receptor. So in the cortex, the signal is mixed, which is also the case for the other SCH23390 radio tracers. So none of them is selective in the cortex. But nevertheless, using this, what we have found is an increase in D1 binding in the dorsolateral prefrontal cortex in schizophrenia that predicted working memory performance. Now, in replicating this, our attempt to replicate came with slightly different results, where the increase in D1 was detected only in the drug-naive patients. And patients that had been previously treated, they all were drug-free for the study. But having received the exposure prior to that, D1 receptor was normal. Could think maybe had normalized. Now there's some data in monkeys to support the concept that D2 blockers can lower D1 levels. This was work, I think, from Pat Gomer-Akik's lab. So this is what we had found. Now, on the other hand, using SCH23390, studies had shown either a deficit, a decrease, no change, or even an increase depending on the medication status. So not terribly informative, and we were interested in dopamine release per se. So we moved on to use a new paradigm to look at dopamine in the cortex. Now I have to tell you a little bit that this property of using a challenge to look at the amount of dopamine release with amphetamine Well, you can have a readout depending on where your D2 radio tracer is giving you a signal. So when you use something like raclopride, the only signal you have is in the striatum. So you could use raclopride to look at dopamine release in the striatum. If you want to look at other at the cortex, it's not very easy. Falipride, which has higher affinity for D2 and can detect D2 outside of the striatum, still doesn't have enough of an affinity to detect the cortex. So studies looking at falipride displacement by stress or cognition or amphetamine in the cortex, I don't believe that. There's just not enough signal. You barely have enough of a signal with FLB. I see some smiles. I'm skeptical. Let me just put it in a milder way. But FLB 457 allows us enough of a signal and is still weak and is still not the most robust way I would want to do imaging, but that was the only thing that was possible. Allows us to image dopamine release in the cortex. Unfortunately, because of its high affinity, it doesn't equilibrate in the striatum during the experiment because there's so many D2 receptors there that the association is still going on and on, doesn't reach the point of equilibrium where you have dissociation and association are equal and you start to see washout. So you don't get to see the washout in the striatum, which is what you need to be able to quantify the signal. So we're restricted to looking at extrastriatal regions with FLB. And doing that, we performed a study looking at FLB before and after amphetamine to image dopamine release in these cortical regions. We also looked at working memory performance using multiple tasks, the letter number sequencing, the NBAG, and using an FMRI paradigm to look at the ability of the DLPFC to activate during working memory performance. We use this complicated task, which is the self-ordered working memory task, which has 8 conditions, because we wanted a very kind of fine-grained way of looking at working memory. And the paper looking at that is currently under review. But basically, this is the task. Subjects see 8 abstract shapes. They first have to choose one shape, and then the shapes kind of like reshuffle. And then they have to choose a second shape, avoiding one that they have previously selected. So they have to keep in mind what is it they've selected to avoid it, to be correct. So as the task increases, as you go from 1 all the way to 8, it becomes more and more difficult to keep track of which shapes have been previously selected. And we basically got a bold fMRI imaging during this working memory task. And a control task where they just have to point at a cross on one of those shapes. The demographics of the samples were 21 controls and 20 patients. They were matched for age, gender, ethnicity, parental socioeconomic status as much as possible, nicotine smoking. We had 6 drug naive and 14 drug-free patients. And they had similar plasma amphetamine levels, which is an important control always, of course. So working memory performance was lower in the patients across all conditions, although both groups' performance kind of dropped down when it got to be at the later steps of the task. So the results of the PET imaging. Now, our main region of interest was the DLPFC. This is what we were interested in, the prefrontal cortex. And using 2 types of outcome measures, the binding potential, BPND, or the volume of distribution, VT, we found the same results, which are the fact that in controls, amphetamine produces a significant decrease in binding, which is what's expected, which is what was found by Raj Narendran when he developed this paradigm that we adopted and basically looked at the test reliability of the amphetamine effects in controls. You do have a small displacement, 7 to 10%, but it's there. Amphetamine produces this effect. Now, this was not there in patients. We did not see an effect of amphetamine. Basically, they did not budge. The binding potential or the volume of distribution are the same pre and post amphetamine. And the differences between groups were significant. And this was not explainable by differences in volume of distribution of the non-displaceable, the reference region, or the virtual reference region. And so this was related to the specific binding of the radiotracer. This is a scatter plot of the displacement in the dorsolateral prefrontal cortex. This is in controls, so on average about 7 to 10. And this is in patients, on average 0%. Now you see always the spread, and this spread is something that we see always in imaging studies. So as a group, they're lower, but there's a lot of variability, and this is variability that we need to understand, and we have some ideas as to how to go forward with this. In terms of the activation related to the task, so this is the average activation. There is like a more sophisticated analysis looking at the inverted U shape of the activation across different steps that I'm not talking about, but some of my people are doing this analysis. Would be better to summarize. But basically, you see that this task really activates or deactivates what they should be doing in terms of the working memory type of signature. And this is not the case in patients that were in the study. And this is the relationship that we found between dopamine release and the activation during the task compared to the control. So what we find is that both groups have the similar relationship, although the patients are kind of traveling lower here, but they have a similar relationship between the capacity to activate and their dopamine capabilities, their dopamine release capabilities. So more dopamine, better activation. So basically it suggests that dopamine is kind of has an important facilitating effect in terms of the DLPFC being able to activate during working memory tasks. We also found this relationship that we found across all our working memory tasks. The letter number sequencing, the SOT, and the NBAC. And this is here just kind of looking at one step during the SOT, which is that in healthy controls, but not in patients, there was a relationship between D2 numbers in the DLPFC and working memory performance. And so this was kind of puzzling as to why we don't see it in patients. So the way I think about this is that if the system is intact, if all the circuitry elements are there, the GABAergic and the glutamatergic neurons and the dopamine release capability, then how much D2 receptors you have will make a difference in terms of being able to perform better. But in dopamine and schizophrenia, this just isn't the case. It suggests to me that there is just too much going on, and the number of D2 receptors just can't make up for all these issues that are going on in the cortex. You don't have enough dopamine for D2 to matter. It's possible also that we didn't have enough power to detect this, of course, but it's kind of a theme that keeps coming back, where D2 makes a difference in controls, but not in patients. Okay, and this was really the big discovery of this study, which was not intentional. We were looking at prefrontal cortical dopamine, but what we found is that throughout the brain and all the cortical regions, but also in extrastriatal subcortical regions and the thalamus, and more importantly in the substantia nigra and VTA, there is a deficit in dopamine release in patients. You see here the amount of displacement. In blue are the controls. So displacing, again, a certain range, anywhere from, few percentages to 7 or 10%. In patients, not. So no displacement, meaning this deficit in dopamine is generalized, is global, is everywhere, except in the striatum. So to summarize, this is what we had found previously, this is what we found in this study, but in addition, we find that there is a deficit in release in the thalamus and in the midbrain. And the reason this is puzzling, well, I'll get to that in a minute, is basically where is the excess striatal dopamine coming from? So to summarize, we have shown high D2 stimulation responsible for psychosis in the rostral caudate. Low dopamine is responsible for negative symptoms, or contributes, I should say, to negative symptoms in the Ventralstriatum. Low dopamine in the cortex may lead to altered cortical function because of this effect on activation during challenge. But then to kind of revisit all the pathways that we have talked about, this is where we were at. What we see is that there is a deficit in the mesocortical, the mesolimbic is relatively, neither too increased nor it's slightly increased. The nigrostriatal to the AST is where most of the dysregulation exists, but not so much in the SMST. And the reason I put this in broken line is because I just don't know at this point, and that's another paradox, where is the excess dopamine in the AST coming from? If it's not from excess midbrain activity, because I would have assumed if there was excess midbrain activity that it would kind of be readable with our paradigms through excess somatodendritic release in the midbrain. Now, it may be that it's just so tiny, affecting just one pathway, that overall the global readout is not affected by this, but that's also kind of puzzling as to why it wouldn't even translate as just no change, but would have a deficit. If it's not driven by midbrain, then the excess in the AST could be driven locally, and driven locally by the kind of modulators of terminal dopamine release, such as the cholinergic system, which can affect dopamine release in the striatum. So these are things to kind of think about. Now, I wanted to kind of go to the midbrain a little bit more in details, because at this point we can't ignore the data, especially because Oliver is sitting here. This is for you. That suggests that there may be excess dopamine synthesis in the midbrain. And so I tried to summarize the data as I see it at this point. There have been postmortem studies. 2 studies reported no change in tyrosine hydroxylase. One study showed an increase in tyrosine hydroxylase mRNA. One reported an increase in tyrosine hydroxylase proteins, which is Oliver's study. And one reported a decrease in tyrosine hydroxylase protein and immunolabeling. And this is probably the same cohort, if I read this correctly, from Perez, Costas, and Rice. And their decrease is kind of in the areas that would just kind of fit in with the idea of a decrease in the mesocortical pathway. In terms of imaging, the F-DOPA, there have been 2 F-DOPA studies, one from Kumakura and one from Oliver, both showing increase of DOPA in the midbrain. However, the study of Oliver, I think, has both patients with schizophrenia and prodromes, and also showed that F-DOPA in the midbrain predicts conversion. So now I could think that this is just, possibly data that needs, more studies. On one hand, Kumakura showed higher K loss in the amygdala midbrain, meaning turnover is bigger. So you could possibly think, okay, there's more synthesis, but there is more turnover, so the actual pool is low, and that would fit with our measure, which is looking at the pool that can be released, right? So there could be consistency. With the study of Oliver has shown that the KICR is increased, and one possibility that could be a confounding effect, although I'm not claiming this is the case, could be differences in non-displaceable binding. So now how can we reconcile this? it could be that, you know, F-dopa is measuring synthesis, we're measuring release, and these are differentially perturbed in the illness, and we need to understand better what is the exact cellular mechanism by which this dopaminergic dysregulation in the midbrain or in the striatum is occurring. And it could be that it's just kind of a difference between, because what we measured is in the chronic phase, it could be that early on in the disease there is excess synthesis, possibly excess release that dies out in the chronic phase in the midbrain, and then you have a global picture of deficit. And it could be that, you know, you need to look at a specific midbrain section. So the midbrain at this point is a big open question. And I don't think we have conclusive evidence either way. And I think we need to revisit it. But I wanted to show you some very intriguing, very preliminary data, and I told Oliver this was for him, which is the following. 4 pairs. So this is not something I would show normally, so you're going to have to forgive me, but it was too tempting. We're looking at dopamine release now in the midbrain and other extrastriatal regions with PHNO and methylphenidate in the prodrome. And we have 4 pairs of prodromal patients and their matched controls. And what we see is that, well, we see the increased release in the striatum as predicted, but look at this. We also see an increased release in the midbrain, but not in the thalamus. So to be followed, the suspense is on. You don't understand. This for me is like really exciting, and because it's just like, what is going on? It's just the next question that we need to answer. So it's going to be very interesting to know what will the data show in the midbrain and whether release is affected early on, but then in the chronic stage is no longer excessive. I just wanted to revisit the AST, and I see that this is taking longer than I thought, but I do have a section I can skip, as I had mentioned earlier. So in the AST, I just wanted to summarize at this point what's going on, because thinking about it is going to be very essential for us to understand the cellular mechanisms of the dopamine dysregulation. So first, just to say that there is agreement across modality, which makes it easier. Everybody can agree that there is excess dopamine there. But then what's happening with the AST is that in controls, you have this kind of gradient where the AST has lower release than the VST and the SMST. This is when you look just at controls. You know, you have, let's say, 15% displacement in the VST, SMST, and you have more like 7% in the AST. And it's this gradient that is no longer there in schizophrenia. In schizophrenia, the AST is just at the same level as all the other regions. This is that increase that we pick up. So what could contribute to this? Because if we think about what is it that brings dopamine release in AST, keeps it down in the normal brain, we could think about maybe an alteration in that system that explains the dysregulation in schizophrenia. And this is where we're going, this is what we're thinking about, so I just thought I would share this with you. It's all speculative. But basically, what determines our readout is a combination of how many dopaminergic projections are there in a region capable of releasing dopamine, and where are the D2 receptors along the synapse, because these are the ones that are most sensitive to the dopamine exposure. So really, our PET signal is kind of an artificial signal that is related to the technicalities of how it's done. So if, for example, you have less dopamine release, then you see less dopamine release. But if you have the same dopamine release, but your D2 receptor is not kind of seeing it because it has moved to extrasynaptic areas, and that's not something you can measure with PET because it's an integrated, poor anatomical resolution kind of measure, then you have less impact of dopamine. So one thing that the AST has that we've been kind of thinking about is this special patch matrix ratio. There's an organization within the striatum of patch and matrix, and they're distinguished by kind of acetylcholine contribution so patches have less cholinergic innervation, and since cholinergic innervation increases dopamine release, and the AST has more patch than matrix, it's possible that this is what makes it that the AST has less dopamine to start with. And so what we're thinking is that what we need to look at is there a kind of a developmental alteration in the patch matrix ratio that makes it such that they have maybe a lower ratio, so that now the dopamine release is in register with the rest of the striatum, or a redistribution of their D2 receptor around the synapse. So this is the kind of cellular studies that needs to be done, postmortem tissues or other animal models that, we just kind of spend a lot of time thinking about, but that could possibly bring about local elements to explain the dysregulation in the striatum and in the AST. So now this is the section that I'm going to just skip because we're at 49 minutes, but I'm going to just give you the bottom line on it. So we have this dopamine dysregulation everywhere in the brain. So how do we go from there to symptoms? And the way I'm thinking about this now is that dopamine is a major modulator of synaptic plasticity. Dopamine modulates NMDA-induced synaptic plasticity almost everywhere in the brain. And if you think about this dysregulation of dopamine as a developmental factor that's going on, it's pretty much going to rework the circuitry. We have evidence that it reworks the circuitry at the structural level, and I've showed some of this data in my earlier lecture today, but also at the functional level. So now we're starting to combine studies of connectivity and PET imaging to be able to address this question. And we have some kind of nice illustrations of this concept that altered dopamine can affect circuitry and all its components. And I think this is one way by which it produces a brain that is so abnormal in so many ways that it can explain all the global type of symptoms that we see. So for the Janssen people, dopamine explains everything in schizophrenia. So I'm going to just kind of glance over this. The mice models have shown that having excess D2 in the striatum alters the bridging collaterals between the go and no-go pathway in a very plastic way, so that more fibers go from the go to the no-go pathway and kind of shifts the balance of these two. When you stimulate the go pathway, now you have inhibition instead of stimulation of behavior. This is the work of Christophe Kellendonk. Hopefully someday you'll be able to hear himself present it. Very impressive work. Our own work, one more thing, this allows us to back translate and go back into humans and look at the connectivity, for example, between the AST and the GPE, the globus pallidum external, where these bridging collaterals should be happening. And we would expect that the connectivity would be increased in patients with schizophrenia that have excess dopamine, and then that would change after treatment. So that's what we're doing. And finally, a last piece is that this is very exciting. This is in revision in the Journal of Neuroscience, so hopefully it'll be in press soon, which is that we discovered this relationship between dopamine release and working memory in healthy controls that is mediated by changing the connectivity between networks during working memory tasks. So, there are some networks, we identify them as being sensitive to the cognitive load, so that connectivity changes when cognitive load increases. And this relationship is pretty much affected by dopamine, and I must say the subcortical dopamine. So I'm not going to go into too much details here, just take my word for it because it's not enough time. But dopamine's effects on working memory are mediated by changing the connectivity between these networks. So basically, dopamine is really affecting the connectivity between these circuits during tasks, and that would be one way by which it could be producing these kind of very different features or symptoms that we see. Okay, so my summary and reformulation. First, I want to tell you that our finding of excess in the striatum and deficit everywhere else could be construed as evidence for the dual phenotype. But do you think it's evidence for the dual phenotype yet? I mean, first, of course, we need to replicate it. But the other issue that's very important that I'm just thinking also about is that we don't know if this excess and deficit are present in the same subjects. It's only if they're present in the same subject that you can talk about the dual phenotype. If they're segregated, where you have some patients have deficit and some patients have excess, and as long as you have abnormal dopamine, you just have this altered, connectivity and problems, then it's not really a dual phenotype. It's kind of different phenotypes, right? So we need to know that. So this is the next question we need to answer. Does the same subject show this imbalance, or is it just across populations? If they co-occur, if we have a dual phenotype, which one is primary? What's the role of D2? How is it that we have at the same time presynaptic phenotype and a postsynaptic D2 phenotype? The extending the deficit to other regions has been really something that's very interesting because it brings about the fact that there's global pathology. It can explain why there's social cognition alteration because of the involvement of things like the amygdala or the ventralstriatum or other regions of the cortex, and etc. How does this affect learning, translate into symptoms? That summarizes the point about restructuring the brain through synaptic plasticity, anatomically and also functionally. And, and this is just kind of going back to our treatment. So we have D2 drugs, and that's great because at least we have something that can work. But obviously, this is way too complex to be addressed just by D2 antagonists, and what we need is to be able to affect the circuitry in all these other places and the deficit in these other places. So we need drugs that can augment D1 signaling where it needs to be augmented, or augment D2 signaling where it needs to be augmented, yet keep psychosis under control. So it's a big challenge for the field. And finally, my summary slide, which is in just a review we're writing for the special issue of biological psychiatry, but it's just kind of, I like the aesthetics of it. It tries to summarize too many elements, but basically this is all the regions that are at stake. Maybe here we could dim the light for a second, if we have a second left. Okay, so on top you're seeing the cortex. In green are the pyramidal glutamatergic cells, and you have these GABAergic interneurons. In light, here it's coming out as purple, are the dopaminergic projections. These dopaminergic projections, you'll see them throughout the slides. Now here you have the ventral striatum, the associative striatum, the sensory motor striatum. And you see the gradient. So there's a lot more projections to the sensory motor striatum than there is to anything else, which explains why there is so much more dopamine in the sensory motor striatum. Now what you see here is in controls and here is in patients, basically summarizing. In the cortex, there is very little dopamine floating around. In the ventral striatum, sensory motor striatum, they're kind of equivalent. In the AST, we have this excess dopamine. And at the level of the midbrain, what in our own data in chronic patients is that there is a deficit in dopamine at the level of the midbrain. This is where you have the striato nigro projections that are D1 medium spiny neurons projections. So this is just to summarize the picture at this point, and I want to thank my collaborators as a big group, because we're, yeah, you can put the lights on. So the imagers within my own division and the preclinical people in my Conti Center, also Yale collaborators for some of the imaging that we do at Yale, my funding, the funding capabilities. And I also want to thank the patients who participate in these very heavy-duty invasive studies. And I want to thank you for allowing me to share my data with you today.
Well, thank you so much for an extremely deep, demanding but rewarding lecture. Time for a couple of questions, and I'm going to ask Oliver House to give a vote of thanks. Are there any questions?
We had many earlier, so maybe they're all answered.
I think this one. Use the mic.
Is that better with the mic? Oh, it's
Yeah.
so much better with the mic, isn't it? Love your studies. You know, the PNAS study from 2000, brilliant. But the one thing I love is when you put the data points there for the amphetamine challenge studies, and you put on one of the axes the symptoms, and there's always one or two people whose symptoms get a wee bit better with amphetamine.
This is another paradox, you know, that you're pointing it out. So we've always noticed that. It's always there. And the reason I think it's an interesting thing that's happening there that we don't fully understand is that I had one day this very compulsive rater. And she comes to me, and, you know, she was a very, you know, very careful. And she comes to me and she said, you know, this patient, the weirdest thing happened when he got amphetamine. He said, the voices are silenced. They're gone. And so interesting, you know. Like, so we look at his data, and he had actually the opposite. So after amphetamine, instead of having more dopamine, it was the opposite. So it was like at this end of the curve, where in a way dopamine decreased. So they had lower dopamine, and at the same time, the psychotic symptoms silenced for a while. So it's as if there's something about the system that we're picking out that's not noise. So what is it? I mean, you know, amphetamine works by making the transporter work in reverse. So it's almost, so instead of picking up dopamine, it's kind of leaking it out, right? But it's possible that there is some kind of interaction with abnormal function of the dopamine transporter in these patients where it's producing the opposite effect. It's lowering levels somehow. It's like you have a leaky transporter, and then, I don't know, it's speculative, but I think it's a real thing. It's just one of those things that we need to understand.
Okay, Oliver.
Should I go sit?
Do you want to sit down? So, as Anissa's mentioned, I've worked quite a lot in this field, so I was quite nervous tonight, especially when I saw the title, Paradoxes, because I was rather worried that I'd come here after spending quite a lot of my career researching dopamine to find out that actually it was all wrong, and actually it had nothing to do with schizophrenia, although we'd spent all this time researching it. But what you've seen elegantly tonight is actually that you can't get away from dopamine if you're interested in schizophrenia. And I wanted just to comment on two things in my vote of thanks. And one is the scientific knowledge, and the other is the scientific process. Because I think Professor Abi-Dargham's talk this evening has demonstrated great advances and insights into both. So the first thing is that when Paul Janssen discovered haloperidol in 1958, we had no idea that dopamine was a neurotransmitter. So there is a journey there, and it's worth reflecting that Janssen as a company have made a big contribution to the treatment of schizophrenia by developing first haloperidol and subsequently risperidone. Both of these are on the WHO list of essential medicines. So that's the first evidence that you can't get away from dopamine. But what we have learned from Professor Abi-Dargham's talk is that things are much more complex, and this journey has evolved greatly since the early days of the 50s and 60s. And Professor Abi-Dargham is very modest, and too modest to give you perhaps the flavour of the import of her work, the scientific advances. So one key hypothesis that has been around for decades is this mesolimbic hypothesis of schizophrenia, that it was excess dopamine in the mesolimbic system that was associated with the psychotic symptoms. But you've seen very elegantly tonight that there's evidence that that's disproved. So that's the first nail in the coffin of a great shibboleth in psychiatry. But there's good news, because you've also seen very elegantly that we now have evidence for another hypothesis that has hung around like a bad smell for many years, but now actually we can see evidence that this dual phenotype potentially could exist, that you've got at least evidence for hypodopaminergia in the cortex, and Anissa's shown that this might extend elsewhere as well, but also the excess in the striatum. And finally, of course, that this is specific to particular parts of the striatum. So what we've seen so elegantly tonight is the evidence of the nature and anatomical functional localization of the dopamine system. And you've seen two major hypotheses in psychiatry addressed rigorously. And that brings me on to my final comment, really, which is about the scientific process. And I have learned so much from seeing Anissa's work over the years, and you'll have seen it demonstrated tonight. Her approach is something that I think we can learn a lot from. And she is meticulous. She builds from preclinical evidence and rigorous. And in an era of Twitter and selfies and instant gratification and attraction for novelty, you see the value of pursuing hypotheses and rigorously dissecting evidence and hypothesis to answer questions. And you can see that there are still questions that we've got to answer going forward. So I would like to ask you to join me in thanking Professor Abi-Dargham for what really is a tour de force, both in demonstrating decades of work in advancing knowledge of dopamine in schizophrenia, but also giving us a lesson in the scientific process. So please join me in thanking her. I want to point out two final things, actually. One other thing is, of course, one other lesson from Anissa Abi-Dargham is that having parents that have the sense to name you with letters that begin with the beginning of the alphabet is also something that is advantageous as well for your recognition in papers. And finally, that's a joke. I think it's the quality of the science that shines through. But finally, I want to encourage you all to join us in the Robin Murray Lecture A and B seminar rooms by the Robin Murray Lecture Theatre, where there will be canapes and wine and an opportunity to ask more questions. So you don't often get much from King's tonight. It's your chance to really enjoy it. Thank you.